Good morning, dear cadets. So, finally, you are going to land in the school, and I am extremely happy. And one more thing is there. Yesterday, I have given you the revision video because I was not having time to prepare the fresh video, and I am hoping that before your arrival, I will complete this unit, and the last unit, that is the solid state, physically in your presence I will teach. Okay, so means it is a matter of great happiness, especially for me. It may be for everyone, but for me it is, I am very extremely happy that you will be landed here. Okay, so now oligosaccharide, that is disaccharide, is over. Now polysaccharide, today I will complete the carbohydrate. Okay, so the polysaccharides, as already I have explained you, polysaccharides are those carbohydrates which are made up of so many monosaccharides unit, and all monosaccharide units held together by the glycosidic linkage. Okay, and these polysaccharides, they are very important for animal kingdom as well as plant kingdom. Okay, so they mainly act as a food storage or structural material. Structural material, you might have heard about the cellulose. Okay, plants cell wall is made up of cellulose. Food storage, so our food storage is glycogen, that is polysaccharide. Plants food storage is starch, that is also polysaccharides. So what are the examples of this one polysaccharides? What are the examples? Their examples I am writing here. Example is cellulose, glycogen, starch, gum, dextrins. Okay, basically in your syllabus cellulose, glycogen and starch has been given. Okay, so the first one I will teach starch. Okay, so starch is basically the polymer of alpha D glucose, and the moment I tell alpha D glucose means what is there? At carbon number first of glucose, OH will be downside. OH will be downside. Okay, in Haworth projection formula. Okay, so the means starch is the polymer of alpha D glucose. Okay, they are the storage polysaccharides of plants, and they are the dietary source for the human beings. High content of starch is found in cereals, roots, tubers and some vegetables. Many tubers we are eating or not? Many roots we are eating, many cereals we are eating, all are containing sufficient quantity of starch. Potato you might have heard. What is there in potato? Starch is only there. Okay, so high content of starch is found in these materials. Okay, now let us move towards the very important part of starch. What is that one? Actually, the components of starch. Starch is basically made up of two types of constituents. One is known as amylose, other is known as amylopectin. Amylose constitute 15 to 20% of starch, and it is water soluble part. What is this one? Water soluble part. You might have heard about that starch give blue color with iodine. Okay, in cold, but the moment you heat the starch, now blue color disappear. So actually the amylose is responsible for giving the blue color with iodine solution. Okay, remember in chlorophyll testing we are using this one or not? Iodine solution. Iodine solution we are using. Yes. So this gives blue color. It is the water. Amylose is the water soluble component, and it constitutes 15 to 20% of starch, and it gives, I wrote here, it gives blue color with iodine. Okay, and the most important thing is it is unbranched chain polymer of alpha D glucose. It is unbranched. Remember this one. It is unbranched chain polymer of alpha D glucose. And what would be the number of alpha D glucose? 200 to 1000. Okay, Now in amylose, what kind of glycosidic linkage is there? Now it is proved that they are amylose is the unbranched chain polymer of alpha D glucose, means near about 200 to 1000 alpha D glucose are there. It means what is there? They must be held with the glycosidic linkage. So what kind of linkage would be there? Alpha glycosidic linkage or C1 C4 glycosidic linkage. In amylose this is found, always remember. In amylose C1 C4. C1 means carbon number 1 of one glucose unit and carbon 4 of second glucose unit, they will be held together by glycosidic linkage. I will make the structure also. Let us have some more idea about the amylopectin. Okay, this is the second component of starch. Second component of starch, and it is water insoluble. Remember this one. It is water insoluble. Amylose will give blue color with iodine solution, but since it is water insoluble, it will not give blue color with iodine solution, and it constitute near about 80 to 85% of starch. Means this is the chief constituent of starch. Amylose form only 15 to 20%, and this is forming 80 to 85%. So which is the chief constituent of starch? Amylopectin is the chief constituent of starch. Okay, it is now another difference is also there. It is branched chain polymer of alpha D glucose. Amylose was unbranched chain polymer of alpha D glucose. Means monomer is same only. In case of amylose also alpha D glucose, in case of amylopectin also alpha D glucose, but glycosidic linkage arrangement is different. In case of amylose, what is there? Unbranched chain of alpha D glucose is there. Here branched chain polymer of alpha D glucose. It does not give blue color with iodine solution. Now last point I wrote this side, this is very important. Having two glycosidic linkage, one is C1 C4 and another one is C1 C6 glycosidic linkage. So C1 C4 just like amylose only, but just see, when I will make, then you will come to know C1 6 branching is also there. So when I will explain, then you will come to know. I have to make those glycosidic linkage, okay, for amylose and amylopectin. Remember, just you remember the glycosidic linkage. In amylopectin, two glycosidic linkage is there. One is C1 C4, another one is C1 C6 glycosidic linkage. C1 C4 is responsible for making the straight chain, but C1 and C6 is responsible for branching, and both are there in amylopectin. While in case of amylose, only one glycosidic linkage is there, that is C1 C4. Okay, so by making one, very easily we will come to know about both. Okay, so just now see the glycosidic linkage is amylose. Okay, so what is the glycosidic linkage? C1 C4, and you know amylose is the chain polymer of alpha D positive glucose. Correct? Yes. So just see, this is alpha D glucose, this is also alpha D glucose, this is also alpha D glucose. They are also alpha D glucose only. Okay, I made two rows. Alpha D glucose very easily you can make, isn't it? Alpha means at first carbon atom OH will be down. In beta means OH will be up. Okay, already I have explained their cyclic structure, Haworth projection formula. Okay, you do one thing. C1 C4 means OH of this one and H of this one. Just remove from here also. OH and H. Remove. Here also you can do the same thing. OH and H. Okay. Remove. Here also OH and H. Okay. Condensation. What is happening here? Between two monosaccharide unit, condensation. So if one molecules of water is removed, these two units will be linked together by one oxygen, divalent oxygen. This one. This is divalent oxygen. Here also this oxygen is there. So this is responsible for linkage between two monosaccharide unit. Now we can write like that. Correct? Yes. Here also just see positive sign and this one remove. Between this one and this one oxygen and this oxygen, in between oxygen. Okay. Here also just see O and O. Okay. Now here also it will happen. So just write down here O like that. Here also O like that. This side also just see this O like that. Here also O like that. Okay. Because another will be counted now. Another will be added. And what is the number of alpha D positive glucose? 200 to 1000. Okay. So we can't make 1000 or 100 here. Now 3, 4 only. So now here another will join. Here another will join. This side also. This side also. Both are alpha D positive glucose only. Just you can check. So this is C1 C4. Just see. C1 of this one, C4 of this one. C1 C4. Here also C1 1 2 3 4 5 6. C1 C4. Here also 1 2 3 4 5 6. Okay. So this is C1 C4. No problem at all. Alpha D positive glucose. Alpha D positive glucose. Here also alpha D positive glucose. Okay. No problem. This is amylose. Now let us move toward the amylopectin. So in amylose what is there? Amylose is the chain polymer of alpha D positive glucose. I have shown here. Now do one thing. Just see. Now write down here amylopectin. Here only will tell you the amylopectin. In amylopectin, how many kinds of glycosidic linkage is there? Amylopectin is also branched chain polymer of alpha D positive glucose. That in case of amylose, what was there? Unbranched chain polymer. This is branched chain polymer. How many type of glycosidic linkage is there? C1 C4 glycosidic linkage, glycosidic linkage. Okay. And another one is C1 C6 glycosidic linkage. Okay. You do one thing, very simple. Okay. This is C1 C4. No problem at all. Now C1 of this one and C. Just see. Here what is there? Now put here OH. Okay. Right now. Now OH of this one and H of this one. Okay. Will come out. Condensation, water molecules. So what would be there? Just see here. Now I will show you. Now this is glycosidic linkage. Just check glycosidic linkage. In NCERT book downside 3 they made and here they have made 2. Why do you know? There is no problem at all. So that this one CH2OH they should be here straight. Just see. I will also delete. Just see. Delete this one. Okay. Just see. Now write down here CH2OH only. Now here write OH. So H of OH of this one, or you can take H of this one and OH of this one. Remove. Okay. So after removal, what would be there? After the removal of one water molecules, what would be left here? Now left out thing would be here. Just see here. This is This is glyco. This is C1C6 glycosidic linkage. Okay, and already C1C4 is there. Here C1C4. This is, now suppose we are talking of this one and this one. This is C1C4 glycosidic linkage, and that is C1C6 glycosidic linkage. So branching is there or not? Both are added together. This is one branch, this is one chain, this is another chain. Both are joined together by this one, this glycosidic linkage, C1C6. Okay, so it is easy only, amylose and amylopectin. And this is branch. What is this one? Branch. Earlier in amylose, what was there? Chain polymer. This is branched chain polymer. That is the difference. Amylose, that is unbranched chain polymer of alpha D glucose. Amylopectin, branched chain polymer of alpha D glucose. Both chains are joined together by C1C6 over this one. Now next one is cellulose. Okay, so now next one is cellulose. Please, one more thing about the starch. Starch is non-reducing sugar. So what are the reducing sugar, what are the non-reducing sugar, already I have explained. Am I correct or not? And examples of reducing sugar, what are the examples of reducing sugar? Glucose, fructose, then lactose, maltose, they were the reducing sugar. And what are the characteristics they have got, that also I have explained. Okay. So, and I have also explained why fructose is reducing the tolerance reagent and failing solution despite having ketonic group. That also I have explained. Okay. Here non-reducing sugar, only one example I have given, that was the sucrose. Please add there, starch is also non-reducing sugar. Now let us see the cellulose. What is cellulose? Cellulose is the structural material. Everybody know this one. The plant cell wall is made up of cellulose. Not only this one, the wall of bacteria is also made up of this one only. It is the chief constituent of cell wall. It is the straight chain polymer of beta D glucose. Okay. In case of starch, what was there? Alpha D positive glucose. Here what is there? Beta D positive glucose. So in the previous structure, only one change would be. What is that one? Here at carbon number 1, OH was downside. Now here OH would be first, means upper side. Okay. Rest everything, just see here. OH of this one and H of this one, just remove. So here what would be there? Just see. OH, H and OH. Remove, condensation. So here what would be there? Just see. It would be like that glycosidic linkage. Just see. O, this is glycosidic linkage. C1C4. Okay. Here also what would be there? C1C4. Correct? This is C1 and this is C4. Beta glycosidic linkage. Now this is beta D glucose. This is also beta D glucose. This is also beta D glucose. So at 4 number, downside OH will come. Okay. You can make like that also. Now it was here na. So O and O. Correct? Yes. It is proved. Now better to make like that. Yes. Here also make like that. First write like that, then like that. Okay. This is. So glycosidic linkage present in the cellulose is C1C4 or beta glycosidic linkage. You can check this one. In this way, this is over cellulose. What is the glycogen? It is the animal starch. What is this one? Food storage in animal body. It is called animal starch. Structurally, it is similar with amylopectin, rather more branched. Structurally, it is similar with amylopectin, rather more branched. It is found in liver, it is found in liver, it is found in brain, it is found in muscles, it is found in yeast and fungi. Yeast and fungi, correct? It is found in yeast and fungi also, in addition to the animal. Okay, so glycogen, it is simple, very much similar with the amylopectin, but it is more branched. Okay, now what are the importance? What are the importance? Just see the importance. This one, it is energy giving food, means I am teaching you the importance of carbohydrate. So carbohydrates are energy giving food. Since ancient time in our Vedas, there are so many things which are mentioned, and all those material belongs to the carbohydrate category only. So this is energy giving food. Not only this one, they are basically used as a food storage in the plant. What is their starch is considered to be the food storage. In animal, glycogen is the food storage. They are considered to be the structural material. Cellulose is the chief constituent of cell wall in plant and bacteria. Important raw material for industry. Just see, furniture industry, you might have seen, means whatever chair, table, whatever wooden furnitures, what are there? What are that? Cellulose only. Lacquer, paint industry, they are only present. Ribose, RNA, deoxyribose, DNA. What is ribose and deoxyribose? They are the pentose sugar only. They are carbohydrate only. Okay, ribose and deoxyribose. And ribose is the chief constituent of RNA, and deoxyribose is the chief constituent of DNA. They are, means what we can say, carbohydrate, they are the chief constituent of nucleic acid, that is DNA and RNA. So these are the some of the importance of carbohydrate. Okay, not only this one, just try to understand. ATP you might have heard, adenosine triphosphate, adenosine diphosphate, ADP. Now what are they? What are they? There also ribose is there. Okay, so they are the chief constituent of energy currency, that is ATP. In this way your carbohydrate is completely finished. Now I will teach proteins. Next one, some in-text questions are also there. Okay, so here some in-text questions are there in page number 412. What is this one? 412. Okay, now just see the question number 14.1. See, glucose or sucrose are soluble in water, but cyclohexane or benzene, 6-membered ring compounds, are insoluble in water. Very simple. Glucose and sucrose, they are the polar compound, that is why they are soluble in water. While benzene and cyclohexane, of course they have got 6 carbon atom only, but they are insoluble in water because they are non-polar in nature. So sucrose and glucose solubility in water because of their polar nature. Non-solubility of cyclohexane and benzene because of the because of the non-polar nature. Next question is, what are the expected product of hydrolysis of lactose? So lactose is made up of 2 monosaccharide unit. One is galactose and other one is glucose. If you are writing, see, I wrote galactose and glucose. Which galactose? Beta D positive glucose. Which glucose? Beta D positive. Okay, so if you are writing this one, that would be the blessing in disguise. Suppose you had been in London and somebody asked, where is Bengaluru? Okay, so now if you are telling Bengaluru is one of the important city of India, okay, this is fine answer, no problem at all. But if you will tell Bengaluru is one of the important city of India and that is the capital of Karnataka, so that would be the excellent answer. So same is the case here also. Galactose and glucose, if you are writing, if you will tell which form, beta D, beta form, that would be better answer. Okay, last question is, how do you explain the absence of aldehyde group in the pentacetate of D-glucose? So pentacetate D-glucose, that do not react with the hydroxylamine. Even though in the molecular formula it seems that free CHO group is there, but actually free CHO group, if it would have been, then it should have reacted with hydroxylamine, because hydroxylamine must react with the aldehyde group, means reaction with the hydroxylamine is the characteristics reaction of aldehyde group. Now if free aldehyde group is there, then it should have reacted. It should have reacted. So pentaacetate D-glucose does not react with the hydroxylamine. Now there are certain summary or some important questions are there which must be asked in the examination, or which had been asked in the recent past in the exam. Like sucrose, what are the hydrolysis product of sucrose? The hydrolysis product of sucrose is glucose and fructose. Which fructose? Beta D positive. Which glucose? Alpha D positive. Okay, so you can mention this one. What kind of glycosidic linkage is found in the sucrose? So in the sucrose, basically C1 C2 type of glycosidic linkage is found, and that is alpha beta glycosidic linkage. Alpha means glucose alpha form, beta means fructose beta form. So you can mention this one. This is the better way to tell C1 C2. You should tell the carbon number. Okay. Now maltose, this is also disaccharide. What are the hydrolysis product of this one? Two molecules of glucose. Which glucose? Alpha D positive glucose. What kind of glycosidic linkage? C1 C4 glycosidic linkage is there. That is also known as alpha glycosidic linkage. Lactose, this is also disaccharide. What are the hydrolysis product? Already written. Galactose and glucose. Which galactose? Beta D. Which glucose? Beta D positive. What kind of glycosidic linkage? Beta glycosidic linkage. Beta glycosidic linkage. Beta glycosidic linkage. Which glycosidic linkage? Beta. Now tell the position of carbon. C1 C4. C1 of galactose and C4 of glucose. Just remember this one. C1 of galactose and C4 of glucose. Okay, remember this one. Okay. Now starch hydrolysis product is ultimate hydrolysis product of starch is alpha D positive glucose. Actually when starch is subjected for hydrolysis, what is formed? Maltose is also formed. When maltose is further subjected for hydrolysis, so alpha D positive glucose will be formed. That is why I wrote word here ultimate hydrolysis product. Okay. Now glycosidic linkage. Starch is made up of two constituents, amylose and amylopectin. In amylose C1 C4. Okay. And you know that C1 C4 of this one alpha D glucose only, because its hydrolysis product is alpha D positive glucose. Amylopectin, two glycosidic linkage is there, C1 and C4 and C1 and C6. This is the branched chain polymer of alpha D positive glucose. Amylose is straight chain polymer or unbranched chain polymer of alpha D positive glucose. Two more are there. What is this one? Glycogen. So glycogen is very much similar with the amylopectin. And cellulose, what kind of okay, let me write that one also. Okay, so I added here cellulose. So what are the hydrolysis product of cellulose? Beta D positive glucose. What kind of glycosidic linkage? Beta glycosidic linkage. Okay, C1 C4 glycosidic linkage. Just now I have finished. And remember, cellulose ultimate hydrolysis product is beta D positive glucose. Glycogen, what is the hydrolysis product? Alpha D positive glucose. Ultimate. Okay. Glycosidic linkage just like a amylopectin, C1 C4 and C1 C6. Rather, it is more branched than amylopectin. In this way, your carbohydrate is completely completed. Now we'll teach you the protein, and proteins are made up of alpha amino acids. So proteins are also polymer only, and they are the polymers of alpha amino acids, polyamides. Okay, so now proteins. What are the proteins? High molecular mass high molecular mass complex biomolecules of alpha amino acid. What are the proteins? Entire information is given here. High molecular mass complex biomolecules of alpha amino acid. High molecular mass means their molecular mass is very high. Moreover to this, they are complex molecules of alpha amino acid. We'll explain what is the meaning of alpha amino acids. So this is this line indicates that they are made up of alpha amino acids. So we can tell proteins are the biopolymer of alpha amino acid. Proteins are the biopolymer of alpha amino acids. Okay. Now actually proteins, they are required for growth and maintenance of our body. Just see, in our body there are so many chemical substances and all are made up of proteins only. Enzymes, hormones, enzymes are there, hormones are there. Now moreover to this, antibodies are there, transport proteins, structural material, contractile material, myosin, that is protein which is present in our bones, correct or not? That is contractile protein. Now different kinds of proteins are there, globular protein, structural protein, okay. So we'll discuss in this particular topic only, but remember basically they required for our growth and maintenance of human body. Actually protein word is derived from Greek word proteose. Proteose. What is the meaning of proteose? Prime importance or primary substance. That is why proteins are the primary substance of human body also. Because without the proteins, our body cannot made up of. Everywhere in all the cells proteins are there. Now, you might have heard about the protoplasm. Protoplasm means cytoplasm plus nucleoplasm. Near about 20% proteins are there in the protoplasm. Okay, nucleoprotein, different kinds of things are there. And biocadets, they are very much aware about the importance of protein. So now proteins, if it is subjected for hydrolysis, so peptides will form. When peptides is further subjected for hydrolysis, then alpha amino acid is formed. So what we can say, the hydrolysis ultimate means proteins ultimate hydrolysis product is alpha amino acids. So what we can say, proteins are ultimately made up of alpha amino acids. Alpha amino acid, amino means NH2 group, correct or not? So just see, proteins, they are also known as polyamides. What is the amide? CONH2 is known as amide group. So in the protein, so many CONH2 groups are there, or CONH group are there. That is why they are known as polyamides also. When I will tell you the peptide chain or peptide linkage, that time you will come to know what is the meaning of this one, polyamides. So proteins are known as polyamides because so many CONH group is there, peptide bond is there. Okay. Now we'll start with the amino acids. Amino acids. So just see, amino means NH2 group. Acid in organic chemistry, only COOH group is known as acid, carboxylic acid. And these amino acids are only alpha amino acids. So what is the meaning of alpha amino acids? Just see. With respect to COOH group, NH2 group is bonded with alpha carbon atom. That is why they are known as alpha amino acid. It doesn't mean that only alpha amino acids are there. Beta amino acids are also there. Gamma, delta amino acids are also there. But basically in the proteins, or you can say basically in the nature, alpha amino acids are found. So in the protein we will discuss basically alpha amino acids only. So they are called alpha amino acids because NH2 group is bonded with alpha carbon atom with respect to the COOH group. They are called alpha amino acids because NH2 group is bonded with alpha carbon atom with respect to COOH group. With respect to COOH group. Correct? Now, don't worry, we'll tell you. This is What is the general formula of amino acid? This is general formula, RCHNH2COOH. This is, you can write like that also, RCHCOOH NH2, like that also you can write. This is general formula, means just see, the carbon atom to which COOH group is bonded, that is known as C terminal. What is that one? C terminal. The carbon atom to which NH2 is bonded, that is known as N terminal. What is this one? N terminal. And these terminal will be very important when peptide linkage will be formed. So COOH is known as C terminal, NH2 is known as N terminal. Now you just see, in all amino acids, one R group is must. R means CH3, CH2, CH3. It means alkyl group. Okay. There is a one amino acid, you should remember, glycine, where R is not there. Where R is not there. Okay. That is glycine. Just see. C terminal is there. This is C terminal. What is this one? C terminal. What is this one? N terminal. But just see, here R is not there. In place of R, what is there? H is there. So glycine is the naturally occurring, or you can say the simplest alpha amino acid where alkyl group is not there. Alkyl group is not there. One more important thing, just pay attention toward this carbon atom. This carbon atom, we can write like that. Okay. See, this carbon atom has got 4 valency. 1, 2, 3, 4. 1, 2, 3, 4 are there. See carefully. See carefully. Its one valency is fulfilled by COOH group, second by H, third by NH2, 4 by R. So this is the general formula of all amino acids. So what we can say, all alpha amino acids are optically active because they have got the chiral carbon atom. This is chiral carbon atom or not? But glycine is optically inactive. Optically inactive. Why? Because it does not have chiral carbon atom. Just see, one valency COOH, second NH2, third and fourth by H. So this is optically inactive. Does not have, I can write here, does not have chiral center. Chiral center. Okay. Rest all amino acids, just see, I can write this side, optically active. Optically active due to presence of chiral center. Due to presence of chiral center. Optically active due to presence of chiral center. Okay. Chiral center means chiral carbon atom. So these are the some of the important information regarding amino acids, and this is general formula. Now representation of amino acids, IUPAC nomenclature. So always remember, as compared to IUPAC nomenclature, their trivial nomenclature system or common nomenclature system is very famous, and common nomenclature system is on the basis of their taste and source. Basically, you are very much aware that common system is sourced from their source from which that compound is obtained. Okay, or that amino acid is obtained, like glycine. Glycine means sweet taste. Okay, that is why they are known as glycine. There are so many amino acids. More important information regarding amino acid is, this R group is only different in different amino acids. Are you getting my point or not? This R group only will be different in different amino acids. Rest everything would be common only. Always remember, glycine, lysine, histidine, lysoine. There are so many amino acids and they will be differed by R group only. Okay, so now nomenclature of amino acids. So as usual, there are two system of nomenclature, IUPAC nomenclature. So in IUPAC system, amino acids are known as amino alkane oic acid. Just see here, this is simple one, glycine, NH2CH HCOOH. So just start the numbering from COOH group, 1 2. So 2 amino ethanoic acid. Okay, next one is this one, alanine. So 1 2, already I told you that in glycine H is there, rest other places alkyl group, that is R group is there. So in alanine here CH3 is there. So 1 2 2 3, 1 2 3. So at 2 number carbon NH2 group is there. So 2 amino propane oic acid. Okay, 2 amino propane oic acid. Next one is valine. This is valine. Everything will, already I told you that in amino acid rest everything will be same except R group. Just see here CH3 is there, here 2 CH3 group is there, they are bonded with CH. That is why in NCERT book what they have written? They have written like that, CH, and here what is there? CH3 whole twice. Correct or not? In case of valine. And this CH is only bonded with this ring. Are you getting my point or not? This is only, see, CH3, CH, and it is bonded with main skeleton. In next category, means next amino acid, what would be there? Here would be another CH2, CH3CH, and here CH2. Now this would be bonded with here. You keep on adding this one, and you will get the subsequent member of amino acids. So now just see, 1 2, just see, 1 2 3, correct? This is 3 is there, 1 can be written, 4. So 1 2 3 4. So what would be the name of this one? This is wrong name. 2, this is the common 2 amino. At 2 number carbon atom amino group is there. So 2 amino, then what is there? 3 methyl butanoic acid, acid. 2 amino 3 methyl butanoic acid, 2 amino 3 methyl butanoic acid. This is IUPAC name of valine. Okay, like that leucine is also there, valine, glycine, alanine, valine, leucine, tyrosine, isoleucine, arginine, lysine, glutamic acid, aspartic acid, histidine, so many are there. Okay, so this is all about their IUPAC nomenclature. Next one is the trivial nomenclature system. In trivial nomenclature system, we should name the amino acids on the basis of their source and property. Source and property, under property test will also come. Like glycine, Greek word, glyco means sweet, that is why it is known as glycine. Like that alanine, alanine and valine. Okay, let us do not pay much attention on this one. Just remember glycine, alanine, leucine, valine, isoleucine, arginine, tyrosine, tyrophane, histidine. These are the some of the name. Okay, threonine, glutamic acid. These are the some of the name of amino acid. Okay, so this is all about the nomenclature system. Representation of amino acid. Amino acids can be represented by 2 ways. 3 letter system. Like just see, glycine. What is the spelling of glycine? Glycine. Okay, so first 3 letter, this is known as 3 letter system. So glycine first 3 letter is GLY, so it is understood it is glycine. Alanine, ALA, valine, VAL. Okay, now 1 letter system. So in 3 letter system, what is there? Some problem was there. What is that problem? That is means a very what to say, complex type of things, and they are beyond the scope of our syllabus. So one letter system, like just see, just take the first letter, glycine G, alanine A, valine V, isoleucine I, arginine A, like that you can take. Now there also some problem is there, like arginine is there. So just see here, alanine also first letter is A, only, and arginine also first letter is A. So they switch over to another thing. So in arginine R they have taken. Okay, now it is given one letter system, you can read this one, arginine. Why they took arginine? Just see, A is a common, so leave the A. Otherwise if you will keep A for arginine, so A for alanine is already written here. Okay, so confusion would be there. So they switch over to next letter, like in arginine, just see, arginine. Okay, so A of arginine and A of alanine is similar, so they took R, next letter. So this is one letter system, like that they have told. And moreover to this one, just see, whose A should not be written and whose R should be written. Try to understand which is first in the category of amino acid, their first letter only should be taken, and subsequent amino acids next letter should be taken. Are you getting my point or not? So alanine is first as compared to arginine. So just see, this is not a very important thing. Okay, now there are some most important information. All naturally occurring amino acids are optically active due to presence of chiral carbon atom except glycine. So remember, glycine is exception for so many things. Another important thing, all naturally occurring amino acids belong to capital L series. Now when I taught you carbohydrate, that time what I taught you? D series. Okay, glyceraldehyde was one of the parameter compound, and with respect to second carbon atom of glyceraldehyde, we have compared their configuration with the carbohydrates. Okay, like glucose, fructose. If their OH is similar, means fifth carbon atom OH is similar with the D-glyceraldehyde OH, so they belongs to the D series. Same is the way here also. Here we will compare all amino acids NH2 group with respect to the OH group of glyceraldehyde. So just try to understand here. Glyceraldehyde formula is this one. See, CHO, CH and OH, and here what is there? CH2OH. Correct? Now if OH is left hand side, left, okay, so this is D-glyceraldehyde. What is this one? D-glyceraldehyde. If OH is in left hand side, COH, H, here COOH, and here CH2OH. So what is this one? L-glyceraldehyde. L-glyceraldehyde. Okay, correct or not? You know this one. Now try to understand, when it was studied, so then it is found that NH2 group of all naturally occurring amino acid is in left hand side. Okay, so that means the configuration of alpha amino acid is very much similar with the L-glyceraldehyde. That is why all belongs to the L series. Okay, this is L-glyceraldehyde. Let us take one amino acids, this one only, NH2. Okay, CH, here COOH, and here H. I took first one. NH2 is left side or not? This is left side, and its OH is also left side. Okay, so what is the meaning of D and L series? If given compound configuration match with D-glyceraldehyde, so that is D series compound. If given compound configuration belongs to the L series, L-glyceraldehyde, that belongs to the L series. correct or not? So NH2 group of amino acid is basically present in the left hand side, okay, and that configuration match with the L-glyceraldehyde. That is why all naturally occurring amino acids belongs to the L series. Okay, have you got it or not? So you should also be careful while writing the chemical formula of amino acid. NH2 group should be left hand side to the chiral carbon atom. Okay, just see, this is chiral carbon atom. In glycine, chiral carbon atom is not there. Chiral carbon atom, NH2 is in left hand side. Here also NH2 is in left hand side. Here everywhere NH2 is in left hand side. Have you got it or not? So while writing the formula, be careful. NH2 group should be left hand side of chiral carbon atom, and COOH always should be in right hand side. Means we can say C terminal should be right of chiral carbon atom, L should be left of chiral carbon atom. Okay, in this way the introduction of amino acid is over. Now tomorrow I will teach you classification of amino acid, and you will have a lot of information. Okay, so that's all for today. Thank you.